Dear conference participants, I present to your attention the results of the study of chromosomes of Pelophylax ridibundus in different population systems of southern Ukraine. Southern Ukraine is characterized by unique biological and genetic diversity. It is home to common and rare species. They retain their relict nature and have a special evolutionary mechanism. The hybridogenetic speciation is of particular interest here. This requires a new strategy for biodiversity assessment, conservation, and reproduction. Pelophylax ridibundus is the most widespread type species of amphibian in bodies of freshwater of southern Ukraine. It is one of the parent species of the hybridogenetic complex of green frogs of the genus Pelophylax. The vast majority of pure ridibundus populations are distributed in the southern and southeastern parts of Ukraine. In orphan regions, Pelophylax ridibundus occurs in combination with Pelophylax lesone and the hybrid taxon Pelophylax esculentus. You can see of the maps the distribution of different population systems of green frogs in Ukraine. On the first map, mixed population. On the second map, pure populations. The karyotype ensures the stability of the species. It is important to investigate how karyologically stable this species is in the conditions of the southern Ukraine. There are investable genetics mechanisms that may have great potential for understanding the role of hybridization as a cause of new species. The purpose was to identify specific characteristic at the level of karyotypes and to conduct a comparative morphological analysis of Pelophylax ridibundus chromosomes from different populations of South Ukraine, include the Crimea. 8 individuals of Pelophylax ridibundus were used in the study. For comparison of karyotypes, typical populations of Pelophylax ridibundus were selected. Point 1. Kherson region genetics Genический district Arabatspit. Point 2. Kharkiv region Izum district near Snishkivka village. Point 3. Kherson region Golopristan district near Starosbulivka village. And point 4. Crimea Bakhchisarai district Zalisny village. A total of 49 metaphase plates were analyzed, 20 of which were selected for morphological analysis. The standard karyotype of Pelophylax ridibundus is symmetrical and represented by 2 end chromosomes. The karyotype include 5 pairs large and 8 pairs of small chromosomes. The analysis revealed a certain variability and heteromorphism of individual homologs and chromosome pairs. The chromosomes of ridibundus type population systems are relatively more stable in the quantitative and qualitative terms. More variable chromosomes in the karyotypes of Pelophylax ridibundus of mixed population systems, ridibundus esculentus and ridibundus esculentus lessonem. The slide shows the normal karyotypes of the studied populations. First can be represented by chromosomes of different sizes but the same morphology. Heteromorphism occurs among a group of small chromosomes. Incomplete sets are often found, which is typical for all analyzed populations. Homologs or even entire pairs are more often lost among small chromosomes. Among large chromosomes, homologs of the 5' may occasionally be lost. The chromosomes of ridibundus type population systems are relatively more stable. A 100-degree of chromosome variability is found in mixed population systems. Here you can see the disturbed karyotypes, for example, no homologs on pars 10 and 12 chromosomes. The results of the study suggest then the relative stability of chromosomes of southern Ukraine, Pelophylax ridibundum in pure ridibundus systems indicate a high potential for their resistance to both environmental factors and interspecific genetics balance. This is especially important in terms of preserving and increased genetics biological diversity, which mainly ensures the sustainability of ecosystem function. Thank you for your attention.